Hello, my name is Joan Mannick, and I'm going to talk to you today about work we're doing at Life Biosciences targeting aging biology, excuse me, as a new way to treat aging-related diseases. So I always like to start with this slide because it shows that almost every chronic disease suddenly takes off in incidence right around the time we hit 60 years of age. And the question is, why does this happen? Why does all the diseases occur around the same time, around the same age? And we used to think this is just random wear and tear, our bodies fall apart, and there's really nothing we can do about this. But there's increasing scientific data that this isn't just random wear and tear, that actually there's a shift in our biology that's occurring as we age that's making us permissive to getting all these various diseases listed on the right. And what's exciting about this is it turns out that the shift in biology is actually modifiable and can be targeted by medicines. And this means that aging is a modifiable risk factor, and this opens up a whole new area of medicine. So, what is this biology that shifts as we age and makes us permissive to getting all those various diseases? Well, there's a discrete number of biological mechanisms that have been identified to date that contribute to why we age. And I'm just going to talk about today about three mechanisms that we're focused on at Life Biosciences and that we're targeting as a new way to treat aging related diseases. So, what are these three mechanisms? The first is mitochondrial uncoupling. So, we're developing safe and innovative drugs that improve metabolism that declines as we age and decrease oxidative stress. The second platform I'm going to talk about is a chaperone-mediated autophagy platform. We're developing therapeutics to prevent the buildup of unwanted proteins that accumulate in aging and disease settings. And the last program is an epigenetic reprogramming platform that's reprogramming the epigenome to a more youthful state as a new way to restore cellular function. So starting with the mitochondrial uncoupling platform, mitochondrial uncouplers are known to increase metabolism, decrease the formation of free radicals, which damage cells, and to extend lifespan in preclinical species. In fact, a previous generation of mitochondrial uncouplers, in particular a drug called DNP, was used in the early 1900s as an effective weight loss drug, but it had unacceptable toxicity. So we've developed a next generation of mitochondrial uncouplers that have mitigated the safety risks of DNP, but still have its benefits of increasing metabolism, which has benefits for obesity and obesity-related disease associated with aging. We've generated 600 novel compounds with key composition of matter, IP granted and filed, excuse me, and in animal models, we've confirmed preclinical safety and efficacy. Specifically, we've shown weight loss in obesity models and reduction of fatty liver and liver inflammation in non-alcoholic steatohepatitis or NASH models. So how do mitochondrial uncouplers work? Well, normally when we eat, the nutrients are oxidized inside mitochondria to generate ATP, which is the main cellular fuel used by cells. So the way this works is when nutrients are oxidized, hydrogen ions are pumped from the mitochondrial matrix into the intermembrane space. And as they accumulate in the intermembrane space, they move back down their concentration gradient into the matrix, and this provides the fuel to generate ATP. What uncouplers do is they allow these hydrogen ions to leak back into the matrix independent of ATP production. So you have to burn more nutrients and more calories to pump enough hydrogen ions into the matrix to provide fuel for ATP generation. So basically, mitochondrial uncouplers force cells to burn more calories to generate cellular fuel. And compounds that cause more calories to be burned obviously will have benefit for obesity. So the rates of obesity in the US and across the world continue to rise, and obesity contributes to the pathogenesis of multiple aging-related diseases, including NASH. This is a figure from the CDC just showing the states in dark red where the obesity rates are over 35%. One of the diseases associated with obesity is NASH. So, NASH is caused when fat starts accumulating in the liver. This causes inflammation in the liver and eventually causes liver failure. So, NASH is a major health problem that's facing us around the globe as the obesity epidemic continues to increase. So we look to see if our mitochondrial uncoupler compounds, a tool compound called BAM15, decreases weight gain in mice fed a high-fat Western diet. So you can see here in mice in green fed a normal diet, there's not a lot of increase in fat mass, but in mice in red and blue fed a high-fat diet, there's a large increase in fat mass. This black line indicates the time point at which the mice in blue were started to be fed one of our mitochondrial uncoupler compounds, BAM15. And you can see the mitochondrial uncoupler compound decreased the fat mass in these mice, despite the fact they were continuing to eat a high-fat diet, and then led to no increase in fat mass accumulation. In contrast, the mice without BAM15 who were fed the high-fat diet continued to have an increase in fat mass. What was important about these compounds is although they decreased the gain in fat mass, they didn't decrease lean body mass, which is skeletal muscle mass. So you can see the skeletal muscle mass maintained itself in the mice who are treated with BAM15, and this was quite different than the fat mass decrease. We also saw that the mice who were fed the high-fat diet had an increase in glucose levels. This is a glucose tolerance test, and the red shows the mice in the high-fat diet had increased glucose levels, but these glucose levels were brought back down to normal in the mice fed BAM15. When we looked at triglyceride levels in blood, in mice fed a chow diet, a normal non-high-fat diet, triglyceride levels weren't increased. They were increased in mice fed the high-fat diet, but these were normalized in the mice fed the high-fat diet with the mitochondrial uncoupler compound. Similarly, liver triglycerides increased in mice fed the high-fat diet, but when they were fed the high-fat diet with BAM15, these liver triglyceride levels were brought down to normal. This can be seen with the red staining of fat in the high-fat diet, in the liver of mice fed a high-fat diet, and this decrease in the red staining of fat in the mice given BAM15. So we're moving this platform forward in NASH and obesity. So moving to the second platform, this is the chaperone-mediated autophagy platform. Chaperone-mediated autophagy, or CMA, is a key mechanism by which cells remove unwanted and toxic proteins. CMA activity declines during aging and in certain diseases due to reduced expression of a particular protein that plays a key role in CMA called LAMP2A. We've identified novel compounds that increase the expression of multiple proteins in the CMA pathway, including LAMP2A, and thereby increase CMA activity. And these CMA activator compounds have been shown to have benefit in multiple preclinical models, including Alzheimer's disease and frontotemporal dementia models. This was published in Cell earlier this year, and in stem cell dysfunction, and this was published in Nature. So how does chaperone-mediated autophagy work? Well, certain proteins in our cells express a 5-amino acid sequence that's recognized by a chaperone protein called HSC70. When HSC70 recognizes these proteins, it targets them to lysosomes in the cells, and the proteins with this pentapeptide motif enter the lysosome through a LAMP2A receptor. Inside the lysosome, these proteins are broken down and recycled to cellular nutrients. What's key here is that CMA activity is controlled by the expression level of this protein called LAMP2A. And the problem when we age is the expression of this LAMP2A protein declines. So here you can see in 22-month-old as compared to 3-month-old mice, LAMP2A expression is lower in the old compared to the young mice. Similarly, in 65-year-old versus 25-year-old humans, LAMP2A expression is lower in the old humans as compared to the younger humans. So the problem when LAMP2A expression levels go down, those soluble proteins that are supposed to be broken down don't get broken down, and they start accumulating and forming insoluble aggregates. So as LAMP2A levels decline with aging, CMA becomes dysfunctional. It can't remove soluble proteins. They form insoluble aggregates, and this causes tissue dysfunction. If we can restore levels of LAMP2A in aging organ systems, this can restore functional CMA activity so that soluble proteins are appropriately recycled. This prevents the accumulation of insoluble proteins, and this leads to tissue health. One of the disease states in which accumulation of insoluble proteins leads to disease pathology are the neurodegenerative diseases such as Alzheimer's disease and frontotemporal dementia. So frontotemporal dementia is caused by accumulation of tau due to tau mutations, and this causes dementia. And Anna Maria Cuervo, this is data from her lab, she showed that treatment with one of Life Biosciences' tool CMA activator compounds reduced tau levels and normalized behavior in a mouse model of frontotemporal dementia. So you can see when you stain for tau, which stains in purple in various regions of the brain in control mice, you don't see accumulation of tau because it's removed by CMA. But when you have tau mutations, this leads to an accumulation of this tau protein that isn't effectively removed. When Anna Maria treated these tau mutant mice with one of our CMA activator compounds, you can see that now the mice were able to remove this tau protein and didn't get this accumulation that disrupts neuronal function. She then looked to see if the neurologic function of these mice with the tau mutation was improved when they were treated with the CMA activator compound. And first, I'm going to show you a test of neurologic function called an open field test. And these little black lines are the tracks that a mouse walks when put in an open field. And you can see a normal mouse in an open field will stay at the periphery because they'll know it's dangerous to move into the center of an open field. In the mice with the tau mutation, the frontotemporal dementia mice, you can see they don't remember to stay at the periphery and they walk all over the middle of an open field. However, when they're treated with the CMA activator compound, they remember again to stay at the periphery. So they have better neurologic function as assessed by the open field test. Anna Maria and her colleagues also looked at visual memory. In dark green is the visual memory of a normal mouse. In dark orange is the decreased visual memory of a mouse with frontotemporal dementia. And you can see the visual memory is restored in the mice with frontotemporal dementia treated with the CMA activator compound. So this is exciting. This is a new target for neurodegenerative diseases such as Alzheimer's disease and frontotemporal dementia, and we're moving forward in these indications. But since CMA is a biology that contributes to multiple aging-related diseases, what's exciting is that activation of this process has benefits in multiple different aging-related conditions. So another condition in which we have shown benefit is the dysfunction of aging hematopoietic stem cells. So hematopoietic stem cells are the cells in our body that generate blood cells such as platelets, white blood cells, and red blood cells. And as we get older, the ability of these hematopoietic stem cells to generate new blood cells declines. And part of the reason the function of these hematopoietic stem cells decline is they have decreased LAMP2A levels. Here you can see LAMP2A in a 4-month-old versus a 30-month-old mouse. And because they have decreased LAMP2A levels, they have decreased CMA activity. You can see CMA activity in a 4-month-old versus 12-month-old versus 30-month-old mouse. The 30-month-old mouse has significantly decreased CMA activity. So this decrease in CMA activity contributes to why these hematopoietic stem cells can't make new blood cells as mice get older. So Anna Maria's group looked to see if she treated these old mice with a CMA activator compound, could she restore the function of aged hematopoietic stem cells? And indeed she did. This is a test of the ability of stem cells to make new blood cells. And you can see when hematopoietic stem cells are isolated from vehicle-treated control mice, they have limited ability to make new blood cells. But in the mice treated with the CMA activator compound, the ability to generate new blood cells is restored in the old mice. So this is a program we're moving forward in diseases associated with hematopoietic stem cell dysfunction. And the last platform I'm going to talk about today is targeting another biology of aging, which is drift in the epigenome. So the epigenome consists of chemical modifications of DNA and DNA binding proteins that regulate gene expression. And aging is caused in part by a drift in this epigenome that leads to dysregulated gene expression. We've in-licensed a proprietary gene therapy program that targets the epigenome of older animals to represent the epigenome of younger organisms via expression of 3 Yamanaka factors, OCT4, SOX2, and KLF4, or OSK. It's been known now for years that these Yamanaka factors can reprogram epigenomes to a younger state, but it was never clear if this could be done safely. So what's exciting about this program is that we've seen no adverse safety findings in mice after 15 months of systemic delivery of OSK gene therapy. This program it was work done in David Sinclair's lab and made the front cover of Nature in late 2020, showing the intravitreal injection of OSK gene therapy in mice, increased nerve regeneration following a crush injury of the optic nerve, restored vision in a mouse glaucoma model, and significantly improved vision in naturally aged mice. So this just shows an overview of the epigenome. Again, the epigenome consists of chemical modifications of DNA and DNA binding proteins that turn genes on and off. But unfortunately, as we age, this epigenome drifts, so the wrong proteins or genes are turned on and off. And the goal of our OSK gene therapy is to reprogram the epigenome back to its younger, correct state. One of the benefits of reprogramming the epigenome is that we may be able to regain the regenerative capacity of tissues that are lost as we age. So the central nervous system is one of the first systems to lose regenerative potential. If you injure a neuron in the adult nervous system, it can't regenerate. Whereas if you injure a neonatal or embryonic nerve, you can see it does retain the capacity to regenerate. And the question is, if we reprogram the epigenome of an adult, could we restore this capacity of nerves to regenerate? And to look at this, David and his colleagues crushed the optic nerve of adult mice and looked at their ability to regenerate after being given control gene therapy or being given OSK gene therapy. And you can see in these blue asterisks are the site at which the optic nerve is crushed, and viable neurons are stained in this yellow color. And you can see that in mice, 5 weeks after being given a control gene therapy, they don't regenerate an optic nerve. But 5 weeks after being given OSK gene therapy, you can see there's regeneration of the optic nerve. So this is exciting, but the question is, does this regenerating nerve actually function and restore vision? So to look at this, David looked in a mouse glaucoma model. Glaucoma is caused by an increase in intraocular pressure that damages the optic nerve and causes vision loss. And so David and his colleagues looked at visual acuity in mouse with glaucoma treated with OSK gene therapy. And they looked at visual function 2 different ways, both by electroretinogram, which measures the electrical activity in the retina, and also by a measure of visual acuity. And you can see in light gray here, just looking at electrical activity, there's reduced electrical activity in the retina of the mouse with glaucoma. But electrical activity is restored when they're treated with OSK gene therapy. Similarly, looking at visual acuity, here in gray is the visual acuity of a normal mouse. You can see when a mouse with glaucoma has decreased visual acuity in orange. In a mouse with glaucoma treated with control gene therapy, there's no significant improvement in visual acuity, where there is a significant improvement in visual acuity in mice treated with OSK gene therapy. This is after 4 weeks of treatment And there's even greater restoration of vision after 8 weeks of treatment. This is really exciting because current therapies for glaucoma are just focused on decreasing intraocular pressure. And this does not restore vision. And sometimes it doesn't even prevent the continued loss of visual function. So if this treatment uh, efficacy translates to humans and we can actually restore vision in diseases like glaucoma, this will be a big medical advance for people. So, in summary, targeting the biology of aging is a really exciting way to develop new therapies for aging related diseases. We know aging is the biggest risk factor for almost every chronic disease, but groundbreaking science over the past decade has revealed that this is actually a modifiable risk factor that can be targeted therapeutically as a new way to treat aging related diseases. Life Biosciences is targeting this aging biology and is targeting 3 specific mechanisms that underlie aging biology, which is epigenetic reprogramming, mitochondrial uncoupling, and chaperone-mediated autophagy. And this unique platform approach allows for the potential to prevent, treat, or reverse multiple aging-related diseases. And we're cheering on all our fellow companies also targeting this biology because there's huge potential and a really exciting amount of work that needs to be done that I think will translate to great new therapies. So thank you very much.